Okay. Huh. Who cares, bro? Who cares? Brother, who cares? activity. Hey Sarvajita. Good luck Shri. Good morning Shubham. श्री ने डीपी चेंज कर दिया. सास के क्या ये? डीपी प्रोफाइल पिक्चर बोलते हैं ना. Whatever. Okay, good luck Anirudh. Good luck everyone. Genetic engineering and modern genetics. Modern biotechnology. Genetic engineering and bioprocessing. I'm kind of tired. In a chromosome, there is a specific DNA sequence called the origin of replication. Identification of DNA with the desirable gene, introduction of the identified DNA into the host, and maintenance of introduced DNA. Maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Identification of DNA with the desirable gene. Steps, 3 basic steps in genetically modifying an organism. Identification of DNA with the desirable gene. Identification of DNA with the desirable gene. Introduction of the identified DNA. Introduction of the identified DNA into the host. Okay. First, identify the desirable gene. identification of DNA with the desirable gene, and then it is introduction. Then part of introduction, first identifying, then introduction of the DNA to the host. Mm. Maintenance of DNA and transfer of the DNA to its progeny. Tools for recombinant DNA technology. Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools: restriction enzyme, polymerase enzyme, ligase, ligase, vector, and host organism. Okay, restriction enzyme, molecular scissor, restriction enzyme, molecular scissor, polymerase enzyme. Is it E. coli, a bacterium closely related to Salmonella? It could replicate using the host DNA polymerase enzyme. Okay, replication. For replication, we need DNA polymerase enzyme. For replication, we need DNA polymerase enzyme. What's wrong with my network? Okay, polymerase enzyme, restriction enzyme, polymerase enzyme, restriction enzyme, polymerase enzyme, ligase and vector, along with host organism. 5 tools of recombinant DNA technology. 5 tools of RDT: vector, host, molecular scissor, DNA ligase, DNA polymerase enzyme, polymerase enzyme, ligase, vector, host, restriction enzyme, polymerase enzyme, and ligase, along with vector and host. These are the tools of recombinant DNA technology. These far are the tools of recombinant DNA technology. Restriction enzyme, molecular scissor, polymerase enzyme, DNA polymerase enzyme, host, vector, and the last one is DNA ligase. DNA ligase. Restriction enzyme. In the year 963, the two enzymes responsible for restricting the growth of bacteria in E. coli were isolated in year 1963. The two enzyme responsible enzyme, okay, two enzyme responsible for restricting the growth of bacteriophage in E. coli were isolated. One of these added methyl group at DNA while the other cut DNA. Okay, one of these added methyl group. Which group? Methyl group DNA while the other cut DNA. The later was called restriction. The later was called restriction endonuclease. Later. Okay. Two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. Okay, two enzymes, 1963. Two enzymes responsible for restricting the growth of bacteriophage in E. coli. Restrict the growth of bacteriophage in E. coli. One of these added methyl group to DNA. Add methyl group to DNA. Add methyl group to DNA, while the other cut DNA. One added methyl group to DNA and the other cut DNA. This was later called restriction endonuclease. The later was called restriction endonuclease. Restriction endonuclease. The first restriction endonuclease was HIND2. First, okay. Restriction endonuclease. Got the DNA. And the first restriction endonuclease was HIND2. First restriction endonuclease. First restriction endonuclease. Got the DNA. HIND2, whose functioning dependent on a specific DNA nucleotide sequence was isolated and characterized 5 years later. Whose functioning depend on a specific DNA nucleotide. Function dependent on a specific DNA nucleotide sequence. Specific DNA nucleotide sequence was isolated and characterized 5 years later. 5 years later. It was found that HIN2 always cut a DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs. How many base pairs? 6 base pairs. Okay. HIND2 always cut DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. It was found that HIND2 always cut DNA molecule at a particular point by recognizing a specific sequence. By recognizing a specific sequence of 6 base pair. By recognizing a specific sequence of अब वो जाएगा वाईफाई जाएगा, लाइट चला गया है ना. It was found that HIND2 always cut DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs. Specific sequence of 6 base pairs. This specific base sequence is known as recognition sequence for HIND2. Beside HIND2, today we know more than 900 restriction enzymes. Beside HIND2, we know 900 restriction enzymes. Today we know more than 900 restriction enzymes that have been isolated from over 230 strains. How many strains of bacteria? 230 strains of bacteria. More than 900 from over 230 strains of bacteria, each of which recognize different recognition sequence. Recognize different recognition sequence. Specific sequence of 6 base pair. That have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequence. The convention for naming this enzyme is the first letter of the name. First letter of the name, genus. Second two letters from species of the prokaryotic cell from which they are isolated. Example, eco R1 comes from Escherichia coli. RY13 eco R1 comes from Net. Intention of naming this enzyme is the first letter of the genus, and the second two letters come from the species of prokaryotic from which they were isolated. Example, eco R1 comes from Escherichia coli. RY13. RY13. In eco R1, the letter R is derived from the name of the strain. Roman number following the name indicates the order. Which enzymes were isolated from that strain of bacteria. Order in which the enzyme were isolated from that strain of bacteria. Order in which the enzyme were isolated from that strain of bacteria. Eco ry13 eco ry13 eco ry13 eco ry13 E. Escherichia. CO denotes genus, species. E denotes genus. CO denotes species, and R strain. R is derived from the name of the strain. Roman number following the name indicates the order in which the enzyme were isolated. Order in which the enzyme were isolated from that strain of bacteria. Order in which the enzyme were isolated from that strain of bacteria. Number following the name indicates the order in which the enzyme were isolated from the strain. Order in which the enzymes were isolated from that strain of bacteria. Enzyme belongs to a large class of enzyme called a nuclease. Rest belongs to a large class of enzymes called nuclease. Just enzyme. Okay, large class of enzyme called the nucleases. Restriction enzymes belong to a large class. Restriction enzyme belongs to a large class of enzyme. Larger class of enzyme called nuclease. Nucleases. Nucleases. There are two kinds of nucleases: exonuclease and endonuclease. Exonuclease remove nucleotide from the end of the DNA. Removes nucleotide from end of DNA, whereas endonuclease make cut at specific position. And the nucleus cuts at specific position within the DNA. Okay, removes nucleotide from end of the DNA. Exonuclease removes nucleotide from the end of the DNA, whereas endonuclease cut the DNA at specific points within the DNA. Okay, make cuts at specific position within the DNA. Each restriction endonuclease, okay, functions by inspecting the length of DNA sequence. Each restriction endonuclease, हरेक restriction endonuclease function काम गर्छ inspecting the length of DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut. First it will bind to the DNA and cut each of the two strand of the double helix at a specific point at their sugar phosphate backbone. Once it finds restriction endonuclease. Once it finds its specific recognition sequence, it will bind to the DNA. And cut each of the two strands. Of double helix at specific points. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the sugar phosphate backbone. Finds specific recognition sequence. It will bind to the DNA and make cut at a specific cut each of the two strands of the double helix DNA at a specific point in their sugar phosphate backbone. Cut sugar phosphate backbone. Cut each of the two strands of the double helix at a specific point in their sugar phosphate backbone. 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 Two strands. Each restriction endonuclease recognizes a specific palindrome nucleotide sequence in the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Okay, palindromic nucleotide. Each restriction enzyme recognizes a specific palindromic nucleotide sequence in the DNA. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands. Action of restriction enzyme. A vector DNA, foreign DNA. Okay. The enzyme cuts both the DNA strand at the same site. Okay. For eco R1, it is GAA-TTC. 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 Okay. Here it is GAA-TTC. Between eco R1 cuts the DNA between base G and A. Cuts between G and A. Cuts between eco R1 cuts DNA between base G and A. Okay. G and sticky ends. Eco R1. Okay. Vector DNA, foreign DNA. Eco R1 cuts. Eco R1 is enzyme restriction in the nuclease. Restriction enzyme. Okay. The enzyme cuts both the DNA strand at the same site between G and A. Only when the sequence GAA-TTC is present in the DNA. GAA-TTC. GAA-TTC. Following sticky ends. DNA fragments join at sticky ends. DNA fragments joins at sticky ends. Recombinant DNA. Now it is recombinant DNA. Steps in formation of recombinant DNA by action of restriction endonuclease enzyme eco R1. Steps in formation of recombinant DNA. Formation of recombinant DNA by action of restriction endonuclease enzyme eco R1. By action of restriction endonuclease eco R1. Eco R1. Steps in formation of recombinant DNA by action eco R1. forward. What's the key? Recombinant DNA. Formation of recombinant DNA by action of restriction formation of recombinant DNA by action of restriction endonuclease. By action of restriction endonuclease. Steps in formation of recombinant DNA by action of restriction endonuclease. Enzyme equal. Enzyme codes both DNA strands. Okay. Do you know what palindrome is? A sequence of the base pair when orientation of reading is kept the same. Reading is kept the same. For instance, read the same on the two strands. The following sequence reads same on two strands, 5' to 3' direction and direction. Restriction CTT AAG. Restriction enzyme cut the strand of DNA a little away. Restriction enzyme cut. Okay. Restriction enzyme cuts the strand of DNA a little away from the center of the palindrome site. A little away from the center. Little away from the center of the palindrome site. This is again AR. Okay. AR. Restriction enzyme cuts the strand of DNA a little away from the center of the palindrome. A little away from the center of the palindrome site. Restriction enzyme cuts the strand of DNA a little away from the center of the palindrome site. Restriction enzyme cuts the strand of DNA away from the center of the palindrome site. But cut the strand of DNA a little away from the center of restriction enzyme cuts the strand of DNA a little away from the center of the palindrome site, but between the same bases on the opposite strand. A restriction enzyme cuts the strand of DNA a little away from the center of the palindrome site, but between the same two bases on the opposite strand. But between the same two bases, GE, same two bases on the opposite strand. But between the same two strands on the opposite strand. But between the same two strand. But between the same two strand on the opposite strand. restriction enzyme cuts the strand of DNA a little away from the center of the palindrome site. Between the same two strands on the opposite side. These leave single-stranded portion. These leaves single-stranded portions at the end. This leaves single-stranded portion at the end. There are overhanging stretches called overhanging stretches. This leaves single-stranded portion at the end. Single-stranded portion at the end. These are overhanging stretches called sticky ends on each strand. On each strand, sticky end on each strand. Sticky end on each strand. These are named so because they form hydrogen bond. Sticky ends, overhanging stretches on each strand. These are named so because they form hydrogen bonds with their complementary counterpart. Okay, these are named so because they form hydrogen bond. These are named so because they form hydrogen bond. These are named so because they form hydrogen bonds. Because they form hydrogen bonds. These are named so because they form hydrogen bonds with their complementary counterpart. Complementary counterparts. Because they're complementary counterparts. Complementary counterpart. Energy strength. These are named so because they form hydrogen bonds with their complementary counterpart. They form hydrogen bond with their complementary counterparts. The stickiness of the ends facilitates the action of enzyme DNA ligase. DNA ligase, enzyme DNA ligase. The stickiness of each end facilitates the action of enzyme DNA ligase. Action of enzyme DNA ligase. Action of enzyme DNA ligase. Okay, these are named so because they form hydrogen bonds with their complementary counterpart. These are named so because they form hydrogen bonds with their complementary counterparts. This stickiness of the ends facilitates the action of enzyme DNA ligase. These are named so because they form hydrogen bonds with their complementary counterparts. This stickiness of the end. These are named so because they form hydrogen bonds with their complementary counterparts. The stickiness of the end facilitates the action of the enzyme DNA ligase. so because they form hydrogen bonds with their complementary cut counterparts. The stickiness of the end facilitates the action of the enzyme DNA ligase. These are overhanging stretches called sticky ends on each strand. So these are overhanging stretches called sticky ends on each strand. These are overhanging stretches called sticky ends on each strands. These are named so because they form hydrogen bonds with their complementary cut counterparts. Astaghfirullah. Enzyme DNA, the stickiness of the end facilitates the action of DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA, which are composed of DNA from different source genome. Recombinant molecule of DNA. Recombinant molecule of DNA composed of DNA from different source genome. Source or genome, which are composed of DNA from different source of genome. Restriction endonucleases are used in genetic engineering to form recombinant DNA, which are composed of DNA from different sources. Recombinant DNA are from different sources. From recombinant DNA are from different sources. Restriction endonucleases are used in genetic engineering to form a recombinant molecule of DNA, which are composed of DNA from different sources. Ligands and ligases foreign DNA. Same restriction. Okay. Same restriction enzyme cutting both foreign DNA and vector DNA at specific points. Cut by the same restriction enzyme, the resultant DNA fragment have the same kind of sticky ends, and this can be joined together end to end. Using DNA ligase figure 9.2, diagrammatic representation of recombinant DNA technology, foreign DNA, same restriction enzyme cutting both foreign DNA and vector DNA at a specific point. Okay, ligase join foreign DNA to plasmid, cutting by restriction. Same restriction endonuclease cutting both foreign and vector DNA at a specific point. Ligases join foreign DNA to DNA plasmid, foreign DNA to plasmid. Recombinant molecule transformation. Ligase, okay, cutting a ligase transformation and regeneration. E. coli, cloning, host, cell division, transformation. Cloning host, E. coli, cloning host, okay, and diagrammatic representation of recombinant DNA technology. Then finally we have cloning vector. Then all this. We can do it. You may have realized that normally, unless one cuts the vector at the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Cannot be created. You have realized that normally, unless one cuts the vector and the source DNA, vector and the source DNA with the same restriction enzyme, unless one cuts the vector and source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Recombinant vector molecule cannot be created. Vector and source of DNA. Source DNA cuts the vector and source DNA with the same restriction enzyme. The recombinant vector molecule cannot be created. Separation and isolation of DNA fragment. Separation and isolation of DNA fragment. The cutting of DNA by restriction endonuclease resulted in the fragment of DNA. The cutting of DNA by restriction endonuclease result in a fragment of DNA. These fragments can be separated by a technique known as gel electrophoresis. Fragment can be separated. DNA fragment separation, okay, separated by a technique called gel electrophoresis. Since DNA fragments are negatively charged, DNA fragments, negatively charged molecule, they can be separated by forcing them to move towards the anode. By forcing them to move towards anode. Positive charge. Under an electric field. Under an electric field, forcing them to move towards the anode under an electric field through the medium matrix. Through a medium or matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from algae, seaweed. Okay, natural polymer. It is natural polymer extracted from seaweed. Natural polymer extracted from seaweed. Nowadays, the most common used matrix is agarose, which is natural polymer extracted from seaweed. Natural polymer extracted from seaweed. Most commonly used matrix is agarose. Most commonly used matrix is agarose, which is natural polymer extracted from seaweed. Used matrix is agarose, which is a natural polymer extracted from seaweed. Since DNA fragments are negatively charged molecule, they can be separated by forcing them. Like a molecule, they can be separated by forcing them to move towards the anode under the electric field through a medium matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from seaweed. Natural polymer extracted from seaweed. Okay, natural polymer extracted from seaweed. Under an electric field through a medium matrix. Nowadays, the most common used matrix is agarose, which is a natural polymer extracted from seaweed. Natural polymer extracted from seaweed, agarose. Okay, the DNA fragments separate, resolve according to their size. through sieving effect provided by the agarose cell. Separate results according to their size through sieving effect provided by agarose cell. The DNA fragment separate results according to their size through sieving effect provided by agarose cell. Okay, hence the smaller the fragment size. That was kind of embarrassing, but study right now. uh, it's okay, ma. Hence, the smaller the fragment size, the farther it moves. The smaller the fragment size, farther it moves. Look at the figure 9.3 and guess at which end of the gel the sample was loaded. Method. Well, largest DNA bands, smallest DNA band. The separated DNA fragment can be visualized only after staining DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. You cannot see pure DNA fragment in the visible light and without staining. You can see bright orange colored bands of DNA in a ethidium bromide stained gel exposed to UV light. The separated band of DNA are cut out from the agarose cell. The separated bands of DNA are cut out from the agarose cell and extracted from the cell piece. This step is known as elution. The separated band of DNA are cut out from the agarose cell redalge. Agar, Gelodium and Gracillarium. And extracted from the gel piece. This step is known as elution. Elution is the process of extracting DNA piece from agarose cell. Okay, the separated band of DNA are cut out from the agarose cell, and extracted from the gel piece. This step is known as elution. The separated band of DNA are cut out from the agarose cell. And extracted from the gel piece. The DNA fragment purified in this way are used in constructing recombinant DNA. The DNA fragment purified in this, it's okay, ma, it's okay, don't worry. The DNA fragment purified in this way are used in constructing recombinant DNA. Used in constructing recombinant DNA by joining them with cloning vector. Used in constructing recombinant DNA. A typical agarose cell electrophoresis showing migration of undigested lane 1 and digested set of DNA fragment lane 2 to 4. Digested, undigested, and digested. DNA fragment lane 2 to 4. Cloning vector. You know that plasmid and bacteriophage have the ability to replicate. Plasmid, bacteriophage have the ability to replicate. Plasmid and bacteriophage have the ability to replicate within bacterial cell, independent of the control of chromosomal DNA. Plasmid bacteriophage have the ability to replicate within the bacterial cell independent of the control of chromosomal DNA. 904. Cloning vector plasmid and bacteriophage for bacteria host. Plasmid and bacteriophage for bacteria host. Agarobacterium tumefaciens for plant dicot host. Retrovirus for animal host. Human cells gene therapy. Retrovirus Agrobacterium tumefaciens. Tumefaciens. Agrobacterium. Agrobacterium tumefaciens. Plasmid and bacteriophage for bacterial host. For bacterial host. For bacterial host दुईटा आंसर bacteriophage and plasmid. Plasmid and bacteriophage have the ability to replicate within bacterial cell. This is for bacteria, okay? For bacterial cell. Independent of the control of chromosomal DNA. Independent of the control of chromosomal DNA. Bacteriophages, because of their high number per cell. Bacteriophage, because of their high number per cell, have very high copy number. They have very high copy number, okay? Bacteriophage, because of their high number per cell, have very high copy number of their genome. Copy number of their genome within the bacterial cell. Per cell, high number per cell. Have very high copy number of their genome within bacterial cell. Some plasmids may have only 1 or 2 copies. Some plasmid may have only 1 or 2 copies per cell, whereas others may have 15 to 100 copies per cell. Per cell. Plasmid, okay? High copy number. Very high copy number of the genome within the bacterial cell. Some plasmid may have only 1 or 2 copies per cell. Their numbers can go even higher. Their numbers can go even higher. Number कति बेसी हुन्छ? Okay, कति बेसी हुन्छ? I also don't know, bro. The number can go even higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, okay, alien piece of DNA with bacteriophage, link alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number. We can multiply its number equal to copy number of the plasmid or bacteriophage. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number. It's okay. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its numbers equal to the copy number of the plasmid or bacteriophage. Plasmid DNA. bacteriophage of plasmid DNA, we can multiply its number equal to the copy number of the plasmid equal to the copy number of the plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help easy linking of the foreign DNA. They help easy linking of the foreign DNA and the selection of recombinant from non-recombinant selection. Okay, present are engineered in such a way, vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinant from non-recombinant. They help easy linking of foreign DNA and selection of recombinant and non-recombinant. Selection of recombinant. Selection of recombinants from non-recombinant. Selection of recombinant from non-recombinant. Five, 6, 1250. Okay. Following are the features that are required to facilitate cloning into a vector. Features that are required to facilitate cloning into a vector. Required to facilitate cloning into a vector. Origin of replication, ORI, selectable marker, cloning site. Origin of replication, ORI, selectable marker and cloning site. Features of good cloning vector, origin of replication, selectable marker and cloning site, recognition site, cloning site or recognition site of restriction endonuclease. Origin of replication, ORI. This is a sequence from where replication starts. Sequence from where replication starts and any piece of DNA will link to this sequence. That's okay, right? I guess so. This is the sequence from where a replication starts and any piece of DNA when linked to the sequence can be made to replicate within the host cell. This is the sequence from where replication starts and any piece of DNA when linked to the sequence can be made to replicate within the host cell. This is the sequence from where replication starts and any piece of DNA when linked to the sequence can be made. Okay, replication. This is a sequence from where replication starts and any piece of DNA when linked to the sequence can be made to replicate within the host. Can be made to replicate when linked to the sequence can be made to replicate within the host cell. Within the host cell. This sequence is also responsible for controlling the copy number. This sequence is also responsible for controlling the copy number of the linked DNA. Copy number of the linked DNA. Okay, ORI. Replication start. Controlling the copy number of the linked DNA. Astaghfirullah. So if one wants to recover many copies, of the targeted DNA, it should be cloned in a vector whose origin supports high copy number. So if one wants to recover many copies of the target DNA. Recover many copies of the target DNA. It should be cloned in a vector whose origin support high copy number, controlling the copy number of linked DNA. ORI को दुईटा function छ. Replication start, controlling the copy number of linked DNA. Controlling the copy number of linked DNA by ORI. Selectable marker. Selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformant and selectively permeating the growth of the transformant. Selectable marker. In addition to ORI, in addition to origin of replication, the vector requires a selectable marker. ORI vector requires a selectable marker which helps in identifying and eliminating non-recombinant transformant and selectively permitting the growth of transformant. Transformation is a procedure through which a piece of DNA is introduced in host bacterium. You'll study the process in subsequent section. Transformation is a procedure through which a piece of DNA is introduced in host bacterium. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. You'll study the process in subsequent section. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline or kanamycin, etc. are considered useful selectable marker for E. coli. Useful selectable marker for E. coli. Okay, E. coli को लगी. Ampicillin, tetracycline, chloramphenicol, tetracycline को क्या बोलते हैं? Kanamycin, etc. are considered useful selectable marker. फर्स्ट स्लॉट कंप्लीट हो गया है. ओके, लेकिन मैं ब्रेक नहीं लूँगी. तुम किसी को लेना है ना तो ब्रेक ले सकते हो. मैं सेकेंड स्लॉट स्टार्ट कर रही. Transformation is a procedure through done. Chloramphenicol, ampicillin, tetramycin and kanamycin, etc. are considered useful selectable markers for E. coli. The normal E. coli cell do not carry resistance against any of these antibiotics. Normal E. coli cell do not carry resistance against any of these antibiotics. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetramycin or kanamycin, etc. are considered useful selectable marker for E. coli. The normal E. coli cell do not carry resistance. Normal E. coli cell do not carry resistance against any of this antibiotics. Normal E. coli do not carry resistance against any of this antibiotics. Normally, these genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetramycin or tetracycline or kanamycin. Okay, cloning selectable marker. Ampicillin, chloramphenicol, tetracycline or kanamycin, etc. are considered useful selectable marker for E. coli. The normal E. coli cell do not carry resistance against any of this antibiotic. PBr 322. PBr 322. Origin of replication. ROP. Tetracycline. TETR. Tetracycline. Ampicillin. AMPR. Here we have three set. Hint 2. Hint 2. Hint 3. Okay. Hint 3. Clave 1. Hint 3 and eco R1. Eco R1. PST1. PUV1. RP. PUV2. SL Sal1. BAM. BAMH. BAMH1. BAMH1. BAM1. BAMH1. BAMH1. Tetracycline. Cyclosporin. Hajur dada. Starting. Yes. Anusha katti din bhai dada na dekho pani. ROP, PUV1, PUV2, cell 1 BAMH 1 cla 1 hind 3 eco R1. Ampicillin resistant tetracycline TET, tetracycline resistant TETR, tetracycline ORI. Origin of replication, this is ROP. This is protein that helps in replication. PAMH1 SAL SAL1 SAL1 ROP PUV PST1 PUV1 PST1 PUV1 CLA1 eco R1 and hind2 hind3 aniraj and hind3 la1 eco R1 PUV1 PST1 PUV2 PUV1 PUV1 PST1 PST-PUV PST-PUV E. coli cloning vector PBR322 showing restriction site hind3 eco R1 BAMH1 SAL1 PUV2 PST1 CLA1. Origin of replication and antibiotic resistant gene ampicillin and tetracycline kanamycin. Okay. ROP codes for protein involved in the replication of plasmid. ROP codes for protein involved in codes for protein. ROP codes for protein responsible for restricting the plasmid copy number. Protein responsible for restricting the plasmid copy number. Bolivar and Roderick. These are restriction site. Okay. Restriction site. Restriction site. Restriction site, BAMH1, SAL1, restriction site. Restricting the plasmid copy number. Protein responsible for restricting the plasmid copy number. Restricting, discovered by Bolivar and Roderick. Restriction site, protein responsible for restricting the plasmid copy number. E. coli cloning vector PBR322 showing restriction site hind3 eco R1 BAMH1 SAL1 PUV2 PST1 CLA1. ORI and antibiotic resistance genes AMPR and ampicillin resistant and tetracycline resistant. ROP codes for the protein involved in replication of plasmid. Restricting the plasmid copy. codes or protein. ROP codes or protein involved in replication of plasmid. Replication of plasmid. PBR 322. Cloning site. Cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition site. In order to link alien DNA, alien vector needs to have single, preferably single recognition site. Okay, in order to link alien DNA, vector need to have a very few, preferably single recognition site for the commonly used restriction enzyme. For the commonly used restriction enzyme. In order to link the alien DNA, the vector needs to have very few, preferably single. In order to link the alien DNA, the vector needs to have a very single, preferably single recognition site for the commonly used restriction enzyme. It's okay, ma. It's okay. You'll be fine, right? Cloning site. In order to link the alien DNA, the vector needs to have a very few, preferably single recognition site for commonly used restriction enzyme. Commonly used restriction enzyme. Molecular scissor. Single recognition site for the commonly used restriction enzyme. Presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. Presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. Figure 9.4. Okay. The ligation of alien DNA is carried out at the restriction site present in one of the two antibiotic resistant gene. The ligation of alien DNA is carried out at restriction site present in one of the two antibiotic resistant gene. शायद The ligation of alien DNA is carried out at restriction. The ligation of alien DNA is carried out at restriction site present in one of the two antibiotic resistance gene. The ligation of alien DNA is carried out at the restriction site present in one of the two restriction. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. The ligation of alien DNA is carried out at restriction site present in one of the two antibiotic resistance genes. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at BAMH1 site of tetracycline resistant gene in a vector PBR 322. The recombinant plasmid will lose tetracycline. Okay. Ligate a foreign DNA in BAMH1 site of tetracycline resistant gene of vector PBR 322. The recombinant plasmid, okay, will lose tetracycline resistance. Lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant one. Lose tetracycline resistance due to insertion of foreign tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant one by plating the transformant on tetracycline containing medium. By plating the transformant on tetracycline containing medium. Okay. The transformant growing on ampicillin containing medium are then the transformant growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinant will grow in ampicillin containing medium but not on that containing tetracycline. Okay, recombinant will grow on ampicillin but not on containing tetracycline. But non-recombinant will grow on medium containing both the antibiotics. But non-recombinants will grow on the medium containing both the antibiotics. Medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformant. Antibiotic resistant gene helps in selecting the transformant, whereas the other antibiotic resistance gene gets inactivated due to the insertion of the alien DNA and helps in selection of recombination. Inactivated due to insertion. Alien DNA helps in selection of recombinants. Selection of recombinants due to inactivation of antibiotics is cumbersome procedure because selection of recombinants due to inactivation of antibiotic. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on the two plates having different antibiotics. Selection of recombinants due to inactivation of antibiotics. Selection of recombinant due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been that is PUC8. Other selectable markers have been developed which differentiate recombinants from non-recombinant. Alternate selectable marker have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in presence of chromosemic substrate. Alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromosemic substances. Produce color in the presence of chromosemic substrate. In presence of chromosemic substrate. PUC8 vector. Ampicillin resistant. Gene of interest. Ligation beta galactosidase. Recombinant plasmid. Okay. Ampicillin beta galactosidase. Non-recombinant plasmid. Ampicillin plating. Recombinant transformant with the GOI gene of interest. Recombinant transformant. Colorless colony. Recombinant. Non-transformant will die. Destroyed. Non-recombinant transformant. Beta galactosidase. Lactate. Beta galactosidase. Produce beta galactosidase along with X-gull. Blue colony. Beta galactosidase produce X-gull. Blue colony. In this, a recombinant DNA is inserted within the coding sequence of the enzyme beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. Produce color in presence of chromosemic substrate. Presence in the presence of chromosemic substrate. Produce color. Blue color. Beta galactosidase plus X-gull. Beta galactosidase. Colorless recombinant. Colorless colony recombinant. Recombinant transformant. Non-recombinant transformant. PUC8. To produce color in presence of chromosemic substrate. In this, a recombinant DNA is okay. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of a chromosemic substrate gives blue color. The presence of chromosemic substrate, that is X-gull plus beta galactosidase. Presence of chromosemic substrate gives blue colored colonies. If the plasmid in the bacteria does not have an insert, presence of insert result into insertional inactivation of the beta galactosidase gene, and the colonies do not produce any color. They are colorless and are identified as recombinant colonies. PUC8. Differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. In this, a recombinant DNA is inserted. In this, a recombinant DNA is inserted in this. A recombinant DNA is inserted within the coding sequence of the enzyme beta galactosidase. Enzyme beta galactosidase inserted in a coding sequence of enzyme beta galactosidase. This result into inactivation of the gene for synthesis of this enzyme beta galactosidase, which is referred to as insertional inactivation. The presence of chromosemic substrate gives blue color colonies if the plasmid in the bacteria does not have an insert. Presence of insert result into insertional inactivation of the beta galactosidase gene, and the colonies do not produce any color. These are identified as recombinant colonies. You see it. Ampicillin. Vectors for cloning gene in plants and animals. In plants, dicot plants, it is something A tumefacens. What is it? Agrobacterium tumefacens, and for animals it is retrovirus. Vectors for bacteria is plasmid or bacteriophage. Okay. Vectors for cloning gene in plants and animals. You may be surprised to know that we have learned the lesson of transferring gene into plants and animals. Transferring gene into plants and animals from bacteria and virus, which have been known for ages. Okay. You may be surprised to know that we have learned the lesson of transferring gene into plants and animals from bacteria and virus. Transferring genes into plants and animals from bacteria and virus, which have known this for ages. How to deliver genes to transform eukaryotic cell. How to deliver genes to transform eukaryotic cell and force them to do what the bacteria or viruses want. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals. You may be surprised to know that we have learned the lesson You may be surprised to know that we have learned the lesson of transferring gene into plants and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cell and force them to do what the bacteria or virus want. For example, Agrobacterium tumefaciens, a pathogen for several pathogen for several dicot plant, is able to deliver a piece of DNA known as tDNA. Transfer DNA. tDNA is able to deliver a piece of DNA known as tDNA to transform normal plant cell into a tumor and direct to transform normal plant cell into tumor and direct this tumor cells to produce the chemicals required by the pathogen. Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as transfer DNA tDNA to transform tumefaciens, a pathogen of several dicot. Agrobacterium, for example, Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA. Dicot plant pathogen, okay, it is a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA to transform normal plant cell into tumor. And direct these tumor cells to produce the chemicals required by the pathogens. And direct this tumor cell to produce chemicals required by the pathogen. Similarly, retrovirus in animals have the ability to transform normal cell into cell. A better understanding of the art of delivering genes by pathogen in their eukaryotic host has generated knowledge to transform these tools of pathogen into useful vectors for delivering genes of interest to humans. The tumor-inducing TI plasmid of Agrobacterium tumefaciens has now been modified into cloning vector, which is no more pathogenic to plant, but it is still able to use the mechanism to deliver genes of our interest to the variety of plants. Similarly, retrovirus have also been disarmed, okay, and are now used to deliver desirable gene into animal cell. So once a gene or a DNA fragment has been ligated into a suitable vector, once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into bacterial, plant or animal host where it multiplies. So once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterium. What is this? For example, Agrobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA, transform normal plant into a tumor and direct this tumor cells to produce the chemical required by the pathogens. Many retrovirus in animal have the ability to transform normal cell into cancerous cell. A better understanding of the art of delivering genes by pathogen in their eukaryotic host has generated knowledge to transform this tools of pathogen into useful vector for delivering gene of interest to human. A tumor inducing DI plasmid. A tumor inducing DI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector. Now been modified into a cloning vector, which has which is no more pathogenic to plant, but is still able to use the mechanism to deliver genes. Cloning vector of our interest to variety of plants. Okay. Vectors for cloning genes in plant and animals. Okay. Similarly, retroviruses also have been disarmed and are now used to deliver desirable genes into animal cell. So once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host, where it multiplies. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host where it multiplies. Just sow this and you'll be fine. So once a gene or a DNA fragment has been ligated into a suitable vector, competent host. tDNA, transfer DNA. Tumor inducing plasmid, DI plasmid. Remove tDNA, transfer DNA. Insert gene of interest. Transfer DNA. tDNA. Dicot plant. Tumor. Gene of interest. Tumor inducing plasmid, DI plasmid. Remove tDNA. Remove DI plasmid. Insert gene of interest. Tumor inducing plasmid. Remove tumor inducing. Tumor inducing plasmid. Remove tumor inducing plasmid. Insert gene of interest. Transfer. Gene of interest can form protein. Disarmed pathogen. Bacteriophage. Disarm the vector. Gene of interest. Gene for pathogenicity is removed from viral genetic material, GOI. Gene of interest is inserted in place of it. Okay. Vector back. Bacterial artificial chromosome. Yeast artificial chromosome. Used BAC and YAC. Back and YAC used for transfer and clone very used to transfer and clone very large piece of DNA in bacteria and yeast cell. Tumor, cancerous cell. Competent host. DNA is a hydrophilic molecule, so it can't pass through cell membrane. So how can bacteria take up a recombinant plasmid? Electroporation. Electroporation. Transient pores. Electroporation. Electroporation. Transient pores. DNA is a hydrophilic molecule, so it can pass through cell membranes. So how can bacteria take up a recombinant plasmid? Electroporation. Transient pores. Treatment with divalent ion. Ca2 plus. Ice, heat and shock. Ice, heat, shock, 42 degrees Celsius and again ice. Vector transfer. Plant, gene gun, biolistic. Gold and tungsten bullet. Animal, microinjection using micropipette. Animal, microinjection. Plants, gene gun, biolistic. Vector transfer, okay. Biolistic, gene gun. Animals, microinjection using micropipette. Bacteria take up a recombinant plasmid. Electroporation, transient pores. Treatment with divalent ion, cation, calcium. Ice, heat shock. Ice, vector transfer. Plants, gene gun biolistics. Animals, microinjection using micropipette. Electroporation, with divalent ion, Ca2 plus. During the foreign gene product. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. If any protein coding gene is expressed in a heterologous host, it is called recombinant protein. Batch culture and continuous culture. Batch culture and culturing method. Batch culture and continuous culture. Okay, it is visible. Sure. Competent host for transformation with recombinant DNA. Since DNA is hydrophilic molecule, it cannot pass through cell membrane. Hydrophilic cannot pass through cell membrane. In order to force bacteria to take up the plasmid, the bacteria cell must first be made competent to take up DNA. This is done by treating them with a specific concentration of divalent cation. Treating them with a specific concentration of divalent cation. Divalent cation such as calcium, which increase the efficiency with which DNA enters. The bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cell by incubating the cell with recombinant DNA. treating them with a specific concentration of divalent cation such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell. Recombinant DNA can then be forced into such cells by incubating the cell, which increase the efficiency with which DNA enters the bacterium through pores in its cell. कुछ बोल नहीं गए. Such as calcium, which increase the efficiency with which DNA enters the bacterium through pores in the cell wall. Recombinant DNA can be forced into such cell. Recombinant DNA can be forced into such cell by incubating the cells with the recombinant DNA on ice. Incubating on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA, incubating them, the cells with recombinant DNA on ice. कहाँ गए कॉपी? Ice heat shock 42 degrees Celsius. Force into such cell by incubating the cell with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on ice. These enables the bacteria to take up the recombinant DNA. I think he did a great job. And then putting them back on ice. This enables the bacteria to take up the recombinant DNA. Recombinant DNA. This is not only way to introduce the DNA into host. In a method known as microinjection, recombinant DNA is directly injected into the nucleus. Okay, directly injected. Recombinant DNA is directly injected into the nucleus of animal cell. In another method suitable for plants, cells are bombarded with high velocity microparticles of gold and tungsten. High velocity microparticles, bombarded with high velocity microparticles, gold and tungsten, coated with DNA. Okay, they are coated with recombinant DNA in a method known as biolistic or gene gun. Okay, coated with the DNA in a method. Okay, coated with the DNA. This is not the only way to introduce alien DNA into host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal's cell. In another method suitable for plant, cells are bombarded with high velocity microparticles of gold and tungsten coated with the DNA in a method known as biolistic or gene gun. And the last method used disarmed pathogen vector. Disarmed pathogen vector, which when allowed to infect the cell, transfer the recombinant DNA to the host. Now that we have learned about the tools for constructing recombinant DNA, let us discuss the processes facilitating recombinant DNA technology. Process facilitating recombinant DNA technology. And the last method used, disarmed pathogen vector. Disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Which when allowed to infect the cell, transfer the recombinant DNA into the host. Excuse me. Process of recombinant DNA technology. Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction endonuclease, isolation of desired gene fragment, ligation of DNA fragment into the vector, transferring the recombinant DNA into the host, culturing the host cell in a medium at large scale, extraction of desired product. Let us examine each of the steps in some detail. Isolation of DNA. Fragmentation of DNA by restriction endonuclease. Fragmentation of DNA by restriction endonuclease. Isolation of desired DNA fragment. Isolation of desired DNA fragment. Ligation of DNA fragment into vector. Transferring the recombinant DNA into the host. Culturing the host cell in a medium at larger scale and extraction of the desired product. Okay. Isolation of genetic material DNA. Recall the nucleic acid is the genetic material of all organisms. Nucleic acid is the genetic material of all organisms without exception. In majority of organisms, this is deoxyribonucleic acid DNA. Deoxyribonucleic acid DNA. In majority of the organism, this is deoxyribo. It needs to be pure form. It needs to be in pure form. In order to cut the DNA with a restriction enzyme, it needs to be in pure form, free from macromolecules. Okay. In majority of organisms, this is deoxyribonucleic acid or DNA. In order to cut the DNA with restriction enzyme, it needs to be in pure form. It needs to be in pure form. In order to cut the DNA, in order to cut the DNA with restriction enzyme, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break cell open to release the DNA along with other macromolecules such as RNA, proteins, polysaccharides and lipid. Macromolecules with other macromolecules such as RNA, protein, polysaccharides and also lipids. This can be achieved by treating the bacterial, cell, plant or animal tissue with enzymes such as lysozyme. Bacteria. Cellulase, plant cell. Chitinase for fungal cell. Lysosome for bacteria, cellulase for plant cell and chitinase for fungus. You know that genes are located on long molecule of DNA intertwined with proteins such as histone. The RNA can be removed by treatment with ribonuclease. Whereas proteins can be removed by treating with protease. Ribonuclease. Treatment with protease, histone protein. Other molecules can be removed by appropriate treatment and purified DNA ultimately. Other molecules can be removed by appropriate treatments and purified DNA ultimately. Purified DNA ultimately precipitates out after the addition of chilled ethanol. Chilled ethanol. PYQ chilled ethanol. Okay, addition of chilled ethanol. This can be seen as collection of fine threads in suspension. Fine threads in suspension. This can be seen as collection of fine thread in the suspension. Suspension. DNA that separates out can be removed by spooling. Removal of DNA that has been separated. Spooling, separation of DNA. Spooling, separation of DNA from suspension. Spooling. Cutting of DNA at a specific location. Restriction enzyme digestion are performed by incubating purified DNA molecule with the restriction enzyme. Restriction enzyme digestion are performed by incubating purified DNA molecule with restriction enzyme. At the optimal condition for that specific enzyme, agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion. Restriction enzyme, that is molecular scissor digestion, are performed by incubating purified DNA molecule with the restriction enzyme at the optimal condition for that specific enzyme. Agarose gel electrophoresis is employed to check the progression. Agarose gel electrophoresis is employed to check the progression of the restriction enzyme digestion. DNA is negatively charged molecule, hence it moves towards the positive electrode and node. Figure 9.3. This process is repeated with the vector DNA also. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source of DNA and the cut vector with space are mixed and ligase is added. This results in preparation of recombinant DNA. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. This results in preparation of recombinant. Recombinant DNA. अब ये रिवाइज क्या होगा ना. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector with the space are mixed with ligase as added. The cutout gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. Vector DNA restriction. Cut vector and the cut vector with space are mixed and ligase is added. From the source DNA and the cut vector with space are mixed and ligase is added. The cutout gene of interest from the source DNA and the cut vector with space are mixed and the ligase is added. This results in the preparation of recombinant DNA. Preparation of recombinant DNA. Amplification of gene of interest using PCR. Polymerase chain reaction. PCR stands for amplification of gene of interest using PCR. Polymerase chain reaction. In this reaction, multiple copies of gene of interest is synthesized. Multiple copies of polymerase chain reaction, copies of gene or DNA, multiple copies of gene of interest is synthesized in vitro using two sets of primer. Multiple copies of gene. Small chemically synthesized oligonucleotide that are complementary to the region of DNA. Oligonucleotide, two primer, two set of primer oligonucleotide that are complementary to the region of DNA. And the enzyme DNA polymerase. Oligonucleotide that are complementary to the region of DNA. Reason to be amplified. Gene of interest. Okay, multiple copies of gene of interest. In vitro using two set of primer. Polymerase chain reacts. Each cycle has 3 steps. Denaturation, annealing and extension. Denaturation, primer annealing and extraction of primer. Extension, denaturation, primer annealing. Denaturation, annealing and extension. Extension, denaturation, primer, annealing and extend. Complementary to the region of DNA. And enzyme DNA polymerase. The enzyme extend the primer using nucleotide provide in the enzyme extends the primer using the nucleotide provide in the reaction. And the xenomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. 1 billion copy made. If the process of replication of DNA is repeated many times, segment of DNA can amplify it appropriately billion times. That is 1 billion copies are made. Be amplified to approximately billion times. 1 billion copies are made. Such repeated amplification is achieved by using of thermostable DNA polymerase isolated from bacterium. Thermus aquaticus. Okay. DNA polymerase isolated from bacterium Thermus aquaticus, which remains active during high temperature. Induced denaturation 90-degree. Induced denaturation of double-stranded DNA. An amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Annealing 55 degrees extension. DNA polymerase stack polymerase. Thermus aquaticus bacterium. Thermus aquaticus withstand high temperature. DNA polymerase stack polymerase. Deoxynucleotide. 30 cycles amplified 1 billion times. Amplified 1 billion times. Amplification. Making something stronger or louder. The process of increasing number of copies of gene. Amplification, increasing the number of copies of a gene DNA segment. Increasing number of copies. Increasing number of copies of a gene. Thermus aquaticus denaturation, primer annealing, extension of primer. DNA polymerase isolated from Bacterium thermus aquaticus, which remains active during the high temperature induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. So amplification is achieved by the use of thermostable DNA polymerase, thermus aquaticus tag polymerase. Okay, DNA fragment, if desired, can now be used to ligate with a vector for further cloning. Insertion of recombinant DNA into host cell organism. Insertion of recombinant DNA the host cell organism. Selectable marker. Obtaining of the foreign gene product. Downstream processing. PCR, polymerase chain reaction, denaturation, annealing, and extension. Gene of interest. Gene cloning, few thousand copies. Vector, host, PCR. Okay. Combining gene of interest with vector. Vectors and gene of interest have to be cut with same restriction endonuclease. Vectors and gene of interest are joined by DNA ligase. Vectors and gene of interest are joined by DNA ligase. Vectors and gene of interest have to be cut with same restriction endonuclease. Vectors and gene of interest are joined by DNA ligase restriction endonuclease. 1960. The restriction modification in 1960, the restriction modification system was discovered in E. coli. Methylase restriction endonuclease prokaryotes, nucleus, exonucleus, and endonucleus. Recognition site. Sequence of base pair recognized by recognition. Restriction endonuclease. So sequence of base pair recognized by restriction endonuclease. RS cloning site. Genus, 2 word, species, strain, and first restriction endonuclease enzyme to be isolated from the strain. Order of isolation of the strain. Haemophilus influenza strain. D. Haemophilus influenza. Order of isolation. Palindromic syndrome. Sequence. EcoRV are also produce lantern. EcoRV also produce lantern. Same restriction enzyme is required for cutting gene of interest and plasmid as doing so gave complementary strands of stickins. DNA ligase addition. Cloning vector. Agrobacterium tuberculum for plant. Dichotomous. Bacteriophage. Plasmid for bacteria and retrovirus for animals. The sound. Insertion of recombinant DNA into the host cell organism. There are several methods of introducing ligated DNA into recipient cells. Recipient cells, after making them competent to receive, take up DNA present in its surrounding. Okay, insertion of recombinant DNA into the host cell. Insertion of recombinant DNA into the host cell organism. There are several methods of introducing the ligated DNA into recipient cell. Recipient cells, after making them competent to receive, take up the DNA present in its surrounding. So if recombinant DNA bearing gene for resistance to an antibiotic, example penicillin, is transferred to E. coli cell, the host cell become transformed into ampicillin. Okay, host cell. So if a recombinant DNA bearing gene for resistance to antibiotic ampicillin is transferred into E. coli, the host cell become transformed into ampicillin resistant cells. If we separate the transformed cell on agar plate containing ampicillin, only transformant will grow. Agar plate containing ampicillin. Containing ampicillin. Astaghfirullah, I feel kinda irritated now. If we shred the transformed cell on agar plate containing ampicillin, only transformant will grow. Untransformed recipient cell will die. Untransformed recipient cell will die. Since due to ampicillin resistance gene, one is able to select ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin. The ampicillin resistant gene is in this case is called selectable marker. If we spread the transformed cell on agar plate containing ampicillin, only transformant will grow. Untransformed recipient cells will die since due to ampicillin resistance gene, one is able to select transformed cell in the presence of ampicillin. In this case, is called a selectable marker. Batch culture, continuous culture. Okay. When you insert a piece of alien DNA into a cloning vector, when you insert a piece of DNA, when you insert a piece of DNA into cloning vector and transfer it into bacteria, plant or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce desirable protein. Desirable protein. Hence, there is a need for a recombinant DNA to be expressed. There is a need for a recombinant DNA to be expressed. The ultimate aim is to produce a desirable protein. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Hence, there is a need for recombinant DNA to be expressed. In almost all recombinant technologies, the ultimate aim is to produce desirable protein. Hence, there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate condition. The expression of foreign gene in the host cell involves understanding many technical details. After having cloned the gene of interest, having optimized the condition to reduce the expression of target protein, and having optimized the condition to induce, and after having cloned the gene of interest and having optimized optimize addition to reduce the expression of the target protein. Expression of target protein, one has to consider producing it on large scale. Can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. Any protein encoding gene expressed in heterologous host. Any protein encoding gene is expressed in heterologous host is called recombinant protein. The cell harboring cloned gene of interest may be grown on a small scale in laboratory. The culture may be used for extracting the desired protein and then purifying it by using different separation techniques. May be used for extracting the desired protein and then purifying it by using different separated techniques. And the cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. The cells can also be multiplied in a continuous culture system. The culture may be used for extracting the desired protein and then purifying it by using different separation techniques. May be used for extracting the desired protein. Different separation techniques. The cell can also be multiplied in a continuous culture system. The cells can also be multiplied in a continuous culture, okay, culture system, wherein the used medium is strained out. From one side, while fresh medium is added from other to maintain the cell in their physiologically most active log exponential phase. Fresh medium, continuous addition of nutrition, removal of product plus waste, bacteria, separation, purification, preservatives, growth time, lag log, no plateau is seen. Bacterial waste accumulate, limited nutrition, batch culture. Fresh medium added, continuous addition of nutrition, removal of product plus waste. It is drained out from one side while fresh medium is added from the other to maintain the cell in the physiologically most maybe may not be, who knows. The cells can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while fresh medium is added from the other to maintain the cell in their physiologically most active log exponential phase. This type of culturing method produces a large biomass leading to larger biomass, higher yields of desired protein. Type of culturing method produce larger biomass leading to higher yield of desired protein. Small volume cultures cannot yield appreciable quantities of product. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors, where a large volume 100 to 1000 liters of culture can be processed was okay large volume of culture can be processed was required. Thus bioreactors can be thought of as vessels in which raw materials are biologically converted into specific product. Raw materials thought of a vessel in which raw material are biologically converted into specific product. Vessels in which raw materials are biologically converted into okay, individual enzymes, etc. Bioreactors can be thought of vessels in which raw materials are biologically converted into specific products. Bioreactors can be thought of a vessel in which raw materials are biologically converted into specific products, individual enzymes, etc. Using plant, animal or human cell. Using microbial plant, animal or human cell. A bioreactor provides optimal condition for achieving the desired product. Bioreactor provide optimal conditions for achieving desired product by providing optimum growth condition, temperature, pH. Okay. The bioreactor provide optimal condition. Bioreactor provide for optimal condition for achieving desired product by providing optimum growth condition. Optimum growth condition: temperature 1, pH 2, substrate, salt, vitamins, and oxygen. Temperature, pH, substrate, salt, vitamins, oxygen. Oxygen, vitamins, salt, temperature, pH, substrate. The most commonly used bioreactors are of stirring type. The most commonly used bioreactors, stirring type, common bioreactors. Stirring type, sparse and simple. Sparse is more advantageous. The most commonly used bioreactors are of stirring type, which are shown in figure 9.7. Simple stir tank bioreactor. Acid base for pH control. Buffer. Buffer. B-U-F-F-E-R. Buffer. Okay. Steam for steam for sterilization. Motor. Foam breaker. Flat plate impeller. Culture broth. Nutrition medium. Medium. Culture broth. Sterile ear. To mix oxygen. O2. Stir tank bioreactor through which stir tank bioreactor through which sterile air bubble are sparged. Increase surface area for oxygen transfer. Bubbles dramatically increase the oxygen transfer area. A stir tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor content. Curved base. Cylindrical and curved base. Okay. The stirrer facilitates even mixing and oxygen availability throughout the bioreactors. The stirrer facilitates even mixing. And oxygen availability. The stirrer facilitates. Okay, यही तो क्वेश्चन आया था ना. Stirrer facilitates even mixing and oxygen availability throughout the bioreactor. The stirrer facilitate even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system. Oxygen delivery system, and a foam control system, a temperature controlled system, pH control system, and sampling ports so that small volume of culture can be withdrawn periodically. Sampling port. Small volume of culture can be withdrawn periodically. Gas entrainment. Gas entrainment. Increase the oxygen transfer. Downstream process. After completion of the biosynthetic stage, product has to be subjected through a series of process before it is ready for marketing as a finished product. The process includes separation, purification, and preservatives. Separation, purification, and where is preservative? Which are collectively referred to as downstreaming process. Separation, purification, which are collectively referred to as downstream process. The product has to be formulated with suitable preservatives. Okay. Such formulation has to undergo through clinical trials. As in case of drugs, strict quality control testing for each product is also required. Strict quality control testing for each product is also required. Okay, strict quality control testing. The downstreaming processing and quality control testing vary from product to product. Downstreaming processing and quality control. Upstreaming, expression of DNA to protein, biosynthetic phase, also called upstreaming process. Expression of DNA into protein, expression of DNA into protein, biosynthetic phase, also called upstreaming. Upstream process, also called upstream process. Expression. Downstream. Separation of protein, purify and preservation. Purification, preservation. Separation, purification and preservation. Finally done. Salmonella typhimurium. Okay. First, Herbert Stanley Cohen and Herbert Boyer. Salmonella typhimurium. Let us focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with native plasmid. The construction of first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA. Antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA. Replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer, 1972. By isolating the antibiotic resistance gene by cutting out a piece of DNA from plasmid which was responsible for conferring antibiotic resistance. By isolating the antibiotic resistance gene by cutting out a piece of DNA from plasmid which was responsible for conferring antibiotic resistance. By isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of so-called molecular scissor restriction enzyme. This plasmid DNA acts as vector to transfer the piece of DNA attached to it. Thermus aquaticus, DNA tag polymerase. Aniline, primer 3'53. DNA polymerase, tag polymerase plus deoxynucleotide. Primer on the set. of interest is synthesized in vitro. Two set of primers, small chemically synthesized oligonucleotide that are complementary to region of DNA. Complementary to region of DNA. Oligonucleotide complementary to region of DNA. Complementary to region of DNA and the enzyme DNA polymerase. DNA polymerase and the enzyme DNA polymerase. The enzyme extends the primer using the nucleotide. The enzyme, okay. DNA polymerase. DNA polymerase, tag polymerase. The enzyme extend primer. Enzyme extend primer using nucleotides provided in the reaction. And the genomic DNA is enzyme extends the primer using the nucleotide provided in the reaction and the genomic DNA is template. They were nice. If the process of replication of the DNA is repeated many times, the segment of DNA can be amplified to approximately 1 billion. The enzyme extended the primer using the oligonucleotide, okay? The enzyme extended the primer using the nucleotide provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from bacterium Thermus aquaticus, which remains active during the high temperature induced denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. DNA polymerase, tag polymerase, deoxynucleotide, DNA polymerase, tag polymerase, okay, DNA polymerase, tag polymerase, deoxynucleotide, DNA polymerase, tag polymerase, deoxynucleotide. Extensions and annealing. Extension amplified 1 billion times, 30 cycle, amplified 1 billion times. Extension पर इस Biotechnology principle and processes. Fast mode. 50 questions, okay. Polymerase chain reaction is used in DNA amplification. The ampicillin resistant gene is used as selectable marker to check transformation. Correct. A and R are correct and R is not correct, but R is not the correct explanation of A. Correct. If any protein encoding gene is expressed in a heterologous host is called a recombinant protein. Which of the following is not a component of downstreaming process? Expression. While isolating DNA from bacteria, which of the following enzyme is not used? DNA from bacteria. Deoxyribonuclease. A suitable vector for gene cloning in higher organism is retrovirus, adenovirus, salmonella, typhimurium, retrovirus. Tumor producing plasmid transform plants, animals, bacteria, fungi. TI plasmid, dicot, plants. Agrobacterium tumefaciens. The purpose of a PCR is amplification of gene, cutting of gene, making a copy of amplification of gene. The palindrome are group of letters that forms the dash when read in both direction, forward and backward, same word, alternate word, same word. If you want to recover many copies of the target DNA, you will choose a vector which does not have ORI, which has which whose ORI origin support high copy number. Read the following statement A to D about certain mistakes in two of them. The first transgenic buffalo, Rose, produced milk which was human alpha lactalbumin enriched. Restriction enzymes are used in isolation of DNA from the macromolecules. Downstreaming processes is one of the step of recombinant DNA technology. Na downstream process ko lagi. End product. DNA, okay, downstream. Processes of recombinant DNA technology are isolation of DNA, fragmentation of DNA, fragmentation of DNA by restriction endonuclease, isolation of desired DNA, isolation of desired DNA, ligation of DNA fragment into vector, transferring the recombinant DNA into the host, recombinant DNA into the host, culturing the host cell in medium at large scale and extraction of desired product. Okay, so downstreaming process is one of the step of our DNA technology, correct? Disarmed pathogen vector are also used in transfer of recombinant DNA into the host. Read the following statement A and D about certain mistakes in two of them. Downstream pathogen is one of the steps of our DNA technology, correct? A and B. Cow Rose produced human protein enriched milk. Okay, the milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies, okay, than natural cow milk. Buffalo. Oh, it's not buffalo, it's cow. Rose. Restriction enzymes are used in isolation of DNA from other macromolecules. Treating the bacterial cell plants, animal tissue with enzymes. Isolation of DNA from other macromolecules is achieved by treating bacterial cell, plant or animal tissue with enzymes such as lysozyme, cellulase, and chitinase. Different types of enzymes are used in isolation of DNA from other macromolecules. In blue-white screening, DAS is used in medium. Chromogenic substances. Blue-white screening, okay. PUC8, right? PUC8. Screening. The correct sequence of making a cell competent is treatment with the divalent cation, incubation of cell with the recombinant DNA on ice, heat shock 42 degrees Celsius, placing on ice, heat shock, incubation of cell with recombinant DNA on ice. treatment with divalent cation. Treatment with divalent cation, placing on eyes, incubation of cell with recombinant DNA. Incubation of cells with recombinant DNA. Incubation of cell with recombinant DNA on ice, heat shock. Treatment with divalent cation, placing on ice. The first one is correct. See, done by treating them with a specific concentration of divalent cation. First, treatment with the divalent cation like calcium ion. This is first, okay, divalent cation first, number one. Through the process, okay, recombinant DNA can be forced into cell. Incubating the cell, then incubation of cell with recombinant DNA. Recombinant DNA can be forced into such cell by incubating the cell with the recombinant DNA. This is option 2, second step. Incubating the cell with the recombinant DNA on ice, first ice, right? Followed by placing them briefly at 42 degrees Celsius heat shock. Placing back on ice. Incubation, then incubation of cell. The sticky ends of a fragmented DNA molecule are made of. The sticky ends of fragmented DNA molecule are made up of calcium salt. The sticky ends of fragmented DNA molecules are made up of calcium salt. The sticky ends of fragmented DNA molecules are made up of endonuclease enzyme, methyl group. Sticky ends of fragmented DNA molecule are made up of methyl group. Unpaired bases, what the heck? The sticky ends of a fragmented DNA molecule is made up of the sticky ends of a fragmented DNA molecule. Sticky ends of fragmented DNA molecules are made up of unpaired bases. That was easy, I think. PCR proceeds in DAE extension, denaturation, annealing, and synthesis. Which of the following? Okay, इसको synthesis भी बोल सकते हैं. Which of the following is not a cloning vector? Yak, PBR322, prove. Plasmid vector in DNA recombinant technology means a virus that transfers gene to bacteria, extrachromosomal autonomously replicating circular DNA. Any fragment of DNA carrying desirable gene, sticky end of DNA. Plasmid vector in DNA recombinant technology means in insertional inactivation of beta galactosidase gene, the bacteria in white colonies will have recombinant plasmid. After completion of the biosynthetic stage in bioreactor, the product undergoes separation and purification process, collectively termed as downstreaming processing. The restriction enzyme HIM2 was isolated from the bacteria Haemophilus influenza. The most commonly used bioreactor of the stirring type, the stirrer facilitates oxygen availability. Insect-tolerant gene from Bacillus thuringiensis is introduced using Ti plasmid of Agrobacterium tumefaciens. DNA polymerase is isolated from bacteria Thermus aquaticus. In the isolation of DNA, removal of protein and RNA is carried out by enzyme dash and dash respectively. DNA, removal of protein and RNA is carried. Protease and ribonuclease. Which of the following statement is not correct about cloning vector? ORI is a sequence responsible for controlling the copy number of the linked DNA. Which of the following statement is not correct about cloning vector? ORI is a sequence responsible for controlling the copy number of the linked DNA. Correct. Selectable marker selectively permits the growth of selectable markers selectively permits the growth of non-recombinant non-transformant. Selectable marker selectively permits the growth of non-transformant. Not correct. Second option B. In order to link the alien DNA, the vector needs to have single recognition site for the commonly used restriction enzyme. The ligation of alien DNA is carried out as a restriction at a restriction site present in one of the two antibiotic resistance genes. When the transformed incorrect one, okay, when the transformed cell on agar plates containing ampicillin are spread, both transformed and non-transformed cell will grow. False. Non-transformed will not grow. Restriction enzymes are used in isolation and separation of DNA from macromolecules. Restriction enzymes are used in isolation and separation of DNA from other macromolecules. Restriction enzymes are used in isolation and separation of DNA from other macromolecules. 2 is false again. 2 कहाँ पर है वो सब झूठ है. Downstreaming processing is one of the steps of recombinant DNA technology. Correct. Disarmed pathogen vectors are also used in transfer of recombinant DNA. Okay, 1 and 2 is false. Enzymes are used in an enzyme catalyzing the removal of nucleotide from the end of DNA is an enzyme catalyzing the removal of 8, 9, 10, 11, 12. An enzyme catalyzing the removal of nucleotide from the end of DNA is exonuclease. The sticky end of a fragmented DNA molecule are made up of unpaired bases. What is the main principle behind rDNA technology? Gene manipulation, DNA amplification, density gradient centrifugation, mutation breeding. What is the main principle behind DNA technology, recombinant DNA technology? Gene manipulation, DNA amplification, density gradient. Not this one. Mutational breeding, gene manipulation. In a polymerase chain reaction, after the denaturation step, why the mixture needs to cool down to lower temperature? In a polymerase chain reaction, PCR, polymerase chain reaction, after the denaturation step, why the mixture needs to cool down to lower temperature? To permit specific annealing of the primer, to give a halt to the reaction mixture, to increase the activity of enzyme tag polymerase, to obtain the multiple copies of the DNA, to permit specific annealing of the primer. Short piece of DNA, single-stranded DNA complementary to specific reason. During electrophoresis, DNA fragment would migrate from anode to cathode, from cathode to anode. Electrophoresis, DNA fragment would migrate from dash to dash, right? DNA fragment would migrate from anode to cathode, from cathode to anode, from both the directions. Smaller ones to anode and larger ones to cathode. During electrophoresis, DNA fragment would migrate from anode to cathode, cathode to anode. Done. current level 30. Oh, guys, okay. I need to reach 40 as soon as possible. मेरा बायो टेक्नोलॉजी कंप्लीट हो गया है, तो मैं 10 मिनट का ब्रेक लेती हूँ, ओके। उसके बाद फिर 2 चैप्टर रिवाइज करना है। कौन है? लक्ष्मण गुरुंग। आर यू फ्रॉम नेपाल? नो। क्षेत्री इज द सरनेम वाइडली यूज्ड इन नेपाल। ब्रो, ग्रो अप। सिक्किम भी एग्जिस्ट करता है, यहाँ पर सब लोग नेपाली होते हैं एंड दे आर इंडियन। बाहर घूमो। दीदी आप इतना पढ़ रही हो तो मुझे लग रहा है। मोहम्मद साद, मुझे भी तो डर लग रहा है, तभी तो पढ़ रही हूँ। हाय निधि। I haven't completed zoology. दीदी मैं बाहर जाके आती हूँ, ओके। 20 मिनट दूर देखती हूँ ताकि मेरे आँख को भी ना वो मिले। Human health and disease, bacterial disease, typhoid, pneumonia, diphtheria, plague and dysentery. Typhoid, pneumonia, diphtheria, plague and dysentery. Typhoid, pneumonia, diphtheria, plague and dysentery are the bacterial disease. Typhoid, pneumonia, plague, dysentery, diphtheria. Typhoid, pneumonia, diphtheria, plague, dysentery. Typhoid, salmonella, typhoid fever, sustained fever. Typhoid fever, sustained fever, 39 to 40 degrees Celsius, fever, pain, and cramps along with intestinal perforation. Typhoid, intestinal perforation. Salmonella typhi, 39 to 40 degree, sustained fever, pain and cramps along with intestinal perforation. Pain and cramps along with intestinal perforation. Bacteria resides in small intestine majorly. Okay, from small intestine moves to blood. It spreads to different organs through blood. Spreads to different organs through blood. In severe case, it perforates small intestine and even leads to death. Typhoid. Undigested waste. Undigested waste feces contain this bacteria. Undigested waste feces contain this bacteria. Contaminated water, contaminated food, healthy individual get infected. This route is called fecal-oral route. Confirmation of typhoid vital test. Other symptoms: pain, stomach pain, weakness, headache, constipation, and loss of appetite. Okay, typhoid fever, Salmonella typhi, sustained fever, 39 to 40 degrees Celsius, pain and cramps along with intestinal perforation. Along with intestinal perforation, bacteria resides in small intestine. From small intestine, it moves to blood, spreads to different organ through blood. Severe case, intestinal perforation, perforates small intestine and even leads to death. Undigested waste. Okay, fecal-oral route. Follow person. Confirmation of typhoid vital test, fecal-oral route. Contaminated water, contaminated food, healthy individual get infected. Contaminated water, contaminated food, healthy individual get infected. Contaminated water and food. Other symptoms: stomach pain, cramps, weakness, headache, constipation, and loss of appetite. Loss of appetite, constipation, headache, and weakness. Headache, weakness, constipation, and loss of appetite. Cholera, Vibrio cholera. Vibrio cholera, contaminated food and water, fecal-oral route, diarrhea, rice water feces, okay. Cholera, Vibrio cholera. Pneumonia, droplet or aerosol infection spread through air. Pneumonia spread through air. Causative agent, Streptococcus pneumonia and Haemophilus influenza. Causative agent, Streptococcus pneumonia, Haemophilus influenza. It causes pneumonia and not influenza. Okay, Haemophilus influenza. Causative agent, Streptococcus pneumonia and Haemophilus influenza. Streptococcus pneumonia and Haemophilus influenza. Infect alveoli of lungs. Fluid filled inside alveoli. Droplet aerosol infection spread through air. Causative agent, Streptococcus pneumonia, Haemophilus influenza. Infect alveoli of lungs, fluid filled inside alveoli. Infect alveoli of lungs, fluid filled inside alveoli. Alveoli can't supply oxygen effectively. Therefore, shortage of oxygen leads to gray nail and bluish lips. Gray nail and bluish lips. Fever, chills, cough, and headache. Fever, chills, cough, and headache. Fever, chills, cough, and headache. Pneumonia, droplet aerosol infection straight through air. Causative agent, Streptococcus pneumonia, Haemophilus influenza. Infect alveoli of lungs, fluid filled inside the alveoli. Alveoli can't supply O2 effectively. Therefore, shortage of oxygen leads to gray, nail, and lips, bluish lips. Fever, chills, cough, and headache. Bubonic plague. Black death. Bubonic plague, black death. Bubonic plague, black death vector. Rat fly. Xenopsella, rat fly. Yersinia pestis, bacteria. Yersinia pestis. Yersinia pestis, bacteria. Bubonic plague, black death. Yersinia pestis, bacteria. Carrier, Xenopsella, rat fly. Red spot turns into black. Bubo, lumps formed in armpits. Bubo, lumps formed in armpit. Okay, vector, rat. Now, rat fly. Vector rat fly, which is also known as Yers, Xenopsella. Xenopsella carrier, Xenopsella. Bacteria, Yersinia pestis. Bubonic plague is also caused by bacteria. Yersinia, Yersinia, Yersinia pestis, pneumonia, droplet aerosol infection, fecal-oral route, typhoid. Okay, typhoid, pneumonia. Bubonic plague is also given. Okay, tuberculosis, Mycobacterium tuberculosis, tuberculosis, Mycobacterium tuberculosis, BCG vaccine, Mycobacterium tuberculosis, leprosy, Mycobacterium tuberculosis causes tuberculosis, leprosy, my, Mycobacterium, leprosy, Mycobacterium, leprosy. Okay, diphtheria, bacterium, diphtheria, Corynebacterium, diphtheria, Gulgotu in infants death. Okay, Gulgotu in infants death, swelling in neck, tonsil, pharyngitis. Gulgotu in infants leads to death. Diphtheria, swelling in neck, tonsil, and pharyngitis. Swelling in neck, tonsil, and pharyngitis. Gray membrane inside the throat. DPT vaccine, diphtheria, pertussis, and tetanus. Diphtheria vaccine, DPT vaccine, diphtheria, pertussis, and tetanus. Pertussis, and tetanus. Okay, tetanus. Clostridium tetani, lockjaw, often fatal. Sustained contraction of voluntary muscles. Anti-tetanus serum given to infected individual. Sustained contraction of voluntary muscles. Often fatal. Lockjaw. Tetanus. Clostridium tetani. Diphtheria, Gulgotu in infant. Okay. DPT. Diphtheria, pertussis, and tetanus. Pertussis, and tetanus. Diphtheria, pertussis, and tetanus. Gulgotu. It's okay. Common cold, viral disease, dysentery, bacillus-shaped bacteria, diarrhea, rod-shaped bacteria, dysentery, rod-shaped bacteria, diarrhea, causative agent, Shigella and E. coli. Causative agent, Shigella and E. coli. Bacterial disease. Bacterial disease. Pneumonia. Dysentery. Haemophilus influenza and Salmonella typhilic, typhoid, THYPHOID. Streptococcus pneumonia, Haemophilus influenza, typhoid, Salmonella typhi, dysentery, plague, PLAGU, Dysentery. I'll go to diphtheria. DIPTHERA. Diphtheria. Tetanus. Typhoid. Tetanus. Clostridium tetani. 7 नंबर में. Mycobacterium. Tuberculosis. Tuberculosis. Leprosy. 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 Bubonic plague. Bulb. Lump formed in armpit. Okay, leave it, bro, leave it. क्या ही करोगी? DPT. Astaghfirullah. Diphtheria ना? Diphtheria. Plague. PLA. Pertussis. Diphtheria, pertussis, and tetanus. Diphtheria, pertussis, and tetanus. Okay. Dysentery. Bacillus shaped bacteria. Diarrhea. Okay. Typhoid, cholera, pneumonia, bubonic plague. Okay. Typhoid is bacterial disease. Even pneumonia, typhoid, diphtheria and dysentery. Viral disease, common cold. Rhinovirus. Viral disease, common cold. Rhinovirus. Droplet infection. Directly and indirectly infect by inhaling droplet or transfer by items. Directly and indirect. Direct and indirect infection. Droplet infection. Okay, droplet. That was aerosol. This one, droplet infection. Rhinovirus directly and indirectly infects by inhaling droplets or transfer by items. Upper respiratory tract is affected, not lungs. Okay, in viral common cold, upper respiratory tract is affected, not lungs. Okay, 3 to 7 days. It lasts for 3 to 7 days. Most common infectious disease in men. 3 to 7 days. Last for 3 to 7 days. Droplet infection. Directly and indirectly infect. Upper respiratory. Affects upper respiratory tract. Droplet infection or viral disease. Dengue, chikungunya, smallpox, chickenpox, measles, rabies, 100% fatal. Mumps, infection of parotid gland. Polio, hepatitis B, common cold. Droplet, chikungunya, smallpox, chickenpox, measles, rabies, mumps, polio, hepatitis B, and common cold. These are droplet infection or viral disease. Chikungunya, dengue, mumps, measles, rabies, then HIV, smallpox, chickenpox, mumps, polio, hepatitis B, common cold. Okay. Dengue, chikungunya, smallpox, chickenpox, measles, rabies, mumps, polio, hepatitis, common cold. Okay. Chikungunya, dengue, mumps, measles, chickenpox, smallpox, hepatitis B, common cold. Still do. rabies and polio. Acquired immunity. Acquired after birth. Acquired immunity acquired after birth. Pathogen specific. B and T lymphocyte. Acquired immunity after birth. Okay, acquired after birth. Pathogen specific. B and T lymphocyte. Pathogen specific. B and T lymphocyte. B lymphocyte forms a specific antibody protein against antigen. B lymphocyte forms a specific antibody against antigen. Bacteria, antibody protein, antigens. Mycobacterium tuberculosis, TB का bacteria. Okay. Memory retained previous infection acquired. क्या हो गया है नेटवर्क को? Memory retained previous infection. Primary response slow or less intense. First time for pathogen entering the body. Antibody formed against virus. Primary response slow or less intense. First time for the pathogen entering the body. Antibody formed against the virus. Memory retained previous infection. Primary response slow or less intense. First time for the pathogen entering the body. Antibody formed against the virus. Memory of B and T lymphocyte formed. Secondary or anamnestic response. Second time infection activate intense or fast response. Virus is killed before causing disease. Okay. Memory retained. Acquired immunity acquired after birth. Pathogen specific. Acquired immunity is pathogen specific. Acquired after birth. Acquired after birth. Acquired immunity acquired after birth. Pathogen specific. B and T lymphocyte. B lymphocyte forms specific antibody against antigen. Example, Mycobacterium tuberculosis. Acquired immunity. Acquired after birth. Pathogen specific. It is pathogen specific. This one is pathogen specific. B and T lymphocytes. B lymphocyte forms specific antibody against antigen. Mycobacterium tuberculosis, TB का bacteria. Memory retained, primary response, secondary or anamnestic response. Memory of acquired immunity, humoral immunity HI, cell-mediated immunity. Acquired immunity is of two types: humoral immunity and cell-mediated immunity. Humoral immunity and cell-mediated immunity. Humoral immunity HI, humoral immunity, blood, pathogen in blood. Produced by B lymphocyte. Humoral immunity is produced by B lymphocyte. Antibody in blood, B lymphocyte. Memory B for secondary response, produced by B lymphocyte. Antibody in blood, produced by B lymphocyte. Memory B for secondary response. Memory B for secondary response. Macrophage engulfing the foreign body. Plasma cell is associated. Okay. CMI, cell mediated immunity. Pathogen को kill कर दे. Pathogen inside cell. Produced by T lymphocyte. It is produced by T lymphocyte. Kills the infected cell. Kills cell mediated immunity. Kills the infected cell. T lymphocyte, produced by T lymphocyte, kills the infected cell. T lymphocyte. T helper cell helps B lymphocyte to produce antibodies. T helper cell helps B lymphocyte to produce antibodies. Memory T. Cell mediated ma memory T for secondary response. Humoral ma memory B for secondary response. Organ grafting, graft resection के लिए T lymphocyte responsible है. Organ grafting, resection, organ grafting, graft resection. T lymphocyte is responsible for graft resection. It is T lymphocyte which is uh, T lymphocyte is responsible for graft resection. Tissue matching, blood type matching, and immunosuppressant. Cyclosporin A. Tissue matching, blood type matching. Tissue matching, blood type matching, and immunosuppressant. Immunosuppressant. Cyclosporin A. Found in fungus Trichoderma polysporum. Fungus. Cyclosporin A. Immunosuppressant cyclosporin A, found in fungus Trichoderma polysporum. Fungus, okay. Macrophage, T helper cell, and cytokinin are associated. Macrophage, T helper cell, and cytokinins are associated. In humoral immunity, plasma cells is associated. IgA, A for IgA for body fluid. IgM, largest. IgE, allergic response. IgG, smallest. Cross placenta, most abundant. IgA, body fluid, blood and milk. IgA, body fluid, blood and milk. IgA, body fluid, blood and milk. IgM, largest. IgM is largest. IgE is allergic response. IgG, smallest. Cross placenta, most abundant. Allergy due to release of histamine and serotonin by mast cell, controlled by antihistamine, adrenaline, and steroids. Okay. Acquired immunity. Acquired, AQUIRED, acquired immunity is of two types: humoral immunity and acquired immunity. Acquired immunity, T lymphocyte, B lymphocyte, okay, blood months, okay, T helper cell, helps in production of T helper cell helps in production of antibodies. Antibodies, okay. T helper cell helps in production of antibodies. Antibodies produced, so two types: acquired immunity, okay. Blood months, humoral immunity is in blood. B memory, acquired immunity means memory means, okay. Memory based, primary and secondary both. Memory means memory, primary and secondary response means both. Primary and secondary response. Secondary response is also called anamnestic. Anamnestic, A-N-A-M, N-E-S-T, I say, anamnestic, okay. T helper cell helps in production of antibodies. 400 something था तब. Bubonic plague, two types: acquired immunity, T helper cell, B lymphocyte, memory, okay, blood represent lymphocyte. Blood matching, tissue matching, blood matching, and immunosuppressant. Cyclosporin A from fungus Trichoderma polysporum. Okay, from Trichoderma polysporum. show you. Macrophage T helper cell cytokinin are associated. Cytokinin are associated. Due to release of allergy, due to release of histamine and serotonin, antihistamine, steroid hormone and adrenaline, antihistamine, adrenaline and steroids, controlled by antihistamine, adrenaline and steroids, adrenaline and steroid control allergy, histamine and serotonin by mast cell. Immune system, lymphoid cell, tissue, organ, tissue, lymphoid cell, tissue, organ plus antibody. Antibodies are soluble, soluble molecule, okay? Lymphoid organ, primary and secondary lymphoid organ. Primary lymphoid organ, secondary lymphoid organ. Lymphoid cell, tissue, organ plus antibody, okay? Lymphoid organ is primary and secondary. Primary lymphoid organ, bone marrow and thymus. Primary lymphoid organ is bone marrow and thymus. Two primary organ, bone marrow and thymus. Bone marrow, B lymphocyte matures. All WBCs formed in bone marrow. All white blood cell formed in bone marrow. Okay, thymus, T lymphocyte matures here. This is part of primary lymphoid organ. T lymphocytes matures here. B lymphocytes maturation. All WBC formed in bone marrow. Immature, antigen sensitive, immature, that is antigen sensitive mature lymphocyte matures in lymphocyte organ. Migrate to antigen sensitive mature lymphocyte. To mature antigen sensitive mature lymphocyte matures. Mature lymphocyte. Immature migrates to secondary lymphocyte. Here, lymphocytes interact with foreign antigens. Activated lymphocytes. Okay, secondary lymphoid organ. Here, lymphocytes interact with foreign antigens. Activated lymphocytes. Effector cells. Activated lymphocytes. Effector cell. Lymphocytes interact with foreign antigen. Secondary lymphocyte organ. Here, lymphocytes interact with foreign antigen. Thymus, lobed structure, decrease in size with lobed structure, okay? Thymosin, peptide hormone. Dorsal to sternum location. Secondary lymphoid organ. Okay, secondary lymphoid organ. Here, lymphocytes interact with foreign antigen. Maturation, basically. Migrate to secondary lymphoid organ. Here, lymphocytes interact with foreign antigens. Foreign antigens are interactors. Activated lymphocyte, effector cell. Lymph node, tonsil, spleen, Peyer's patches, malt, appendix. Okay. Lymph node, tonsil, spleen, Peyer's patches, malt, mucosa associated lymphoid tissue, appendix. Okay, these are the five. Secondary lymphoid organ, tonsil, malt, Peyer's patches, spleen, lymph node, appendix. Lymph node, solid structure, small. Okay, lymph node is solid structure, but it is a small. Checkpoint, B and T lymphocyte. It is checkpoint for B and T lymphocytes. Checkpoint for B and T. Lymphoid node. Solid structure, small. Lymph node has B and T lymphocyte. Lymph node has B and T lymphocyte. Checkpoint, tonsil. Spleen, graveyard of RBC, reservoir of RBC, large bean shaped. Spleen is large bean shaped. Large bean shaped. Graveyard of RBC, reservoir of RBC. Lymph node has lymphocyte. Lymph node has lymphocyte. Phagocytes. Filtration of blood, body fluid, trap blood-borne microbes. Lymph node has lymphocytes, phagocytes. Filtration of blood, body fluid, trap blood-borne microbes, Pierce patches, PP. Pierce patches, types of malt, lymphocyte cell, small intestine, malt, mucosa-associated lymphoid tissue, respiratory passes, digestive passes, and urogenital passes. Respiratory passes, digestive passes, and urogenital passes. Malt, mucosa-associated lymphoid tissue, malt. Mucosa secretes mucus. 50% of total lymphoid tissue in body. 50% of total lymphoid tissue present in body. Malt is 50% of total lymphoid tissue present in body. Lymph node, PNT lymphocyte. Solid structure small, okay? SPP. Pierced patches in intestine. Pierced patches. Types of malt. It is a type of malt, okay? Mucosa associated lymphoid tissue. Secrete mucus. Respiratory tract, urogenital tract, and digestive tract. Respiratory passes, digestive passes, and urogenital passes. 50% and urogenital passes. 50% of total lymphoid tissue in body. Tube shape or large intestine. Part of immune system helps fight disease. Okay, appendix. Helper T cell. Activate B and T lymphocyte. Helper T cell. Activate B and T lymphocyte. Production of antibodies. Plasma cell. Activate in production of cells. Combine antigen causing plant cell. Plasma cell. Activate in production antibodies. Killer T cell. Combine with antigen causing lysis and release cytokine. Secondary lymphoid organ.